Hello there, and welcome back to Dreamy Science Stories. I'm really glad you're here tonight. Maybe the day felt a little long, or maybe you simply enjoy drifting into sleep while the world quietly explains itself. For the next little while, you don't need to focus or remember anything. Just let your breathing slow and allow your thoughts to soften. Tonight, we're stepping into a forest that doesn't speak aloud. There are no voices here, no plans, no thoughts in the way humans understand them. And yet, inside every tree, something is always moving. Tiny signals travel through wood and leaf, sliding gently along water-filled pathways, shifting charges across living cells, carrying information without words or awareness. Trees do not think, but they sense. They register change. They respond slowly, patiently, and quietly. This is not a story about forests as networks or systems making decisions together. This is something smaller and much softer. It's the story of how a single tree knows what is happening inside its own body. And if these calm journeys help you unwind, feel free to gently like the video, share it with someone who enjoys quiet science, or leave a soft comment below. It helps others find their way here too. Now, let's begin. Inside every living plant cell, there is a quiet separation. The inside of the cell carries a slightly different electrical charge than the space around it, held in place by a thin, living membrane. This difference is small, almost delicate, but it is carefully maintained as long as the cell remains alive. Nothing about this electrical state feels dramatic. There is no spark, no flash, no sudden burst of energy. It exists quietly, moment by moment, forming a steady background against which change can be felt. The charge is sustained by the uneven distribution of ions. Potassium and calcium move slowly through specialized channels embedded in the membrane, guided by structure rather than intention. Their motion is constant but restrained, maintaining balance instead of seeking speed. Most of the time, these channels remain closed. The electrical potential across the membrane holds steady. The cell carries on with its internal processes, unaware of anything beyond its own boundaries. Then, a disturbance arrives. A leaf bends under pressure. A surface is torn. The membrane deforms slightly, and some ion channels open in response. Begin to flow more freely in that small region of tissue. The electrical balance shifts. This shift does not announce itself. It does not trigger an alarm. It simply alters the local electrical state, creating a difference that did not exist a moment before. From that point, the change begins to spread. It moves from the cell to cell, not because it is directed, but because the cells are connected. Where membranes touch, where fluids pass, where living continuity exists, the electrical change can travel. The signal follows the architecture of the plant. It moves along hydrated tissues, through veins and soft cellular layers that allow ions to influence neighboring cells. There is no central route, only pathways shaped by growth. The movement is slow, far slower than the electrical impulses that race through animal nerves. Minutes may pass as the signal crosses a small portion of leaf, sometimes longer. There is no urgency in this pace. The signal is not meant to arrive quickly. It is meant to arrive intact. As the electrical change travels, its strength diminishes. The difference in charge becomes gentler with distance, fading naturally as ions are redistributed. Nearby, tissues feel the shift clearly. Farther regions experience only a subtle adjustment, or none at all. This fading is not a failure. It is part of the design. By weakening as it spreads, the signal limits its own reach. Only areas close to the disturbance are strongly affected, while distant tissues remain calm, conserving energy. Different kinds of disturbance produce different electrical patterns. A light mechanical touch creates a brief, shallow change in voltage. A tearing injury produces a deeper, longer-lasting shift. The cells do not know what caused the disturbance. They respond only to the pattern they receive. Electrical signals in plants are not fixed in direction. Changes in voltage can propagate forward, slow, or even reverse briefly before returning to baseline. Once ion distributions are restored, the original electrical balance reasserts itself. The system remains flexible. Nothing is locked in place. These signals do not require a nervous system. There are no neurons, no synapses, no specialized cells devoted solely to transmission. Any living cell with an intact membrane and access to ions can participate. The plant's internal structure determines how far the signal can go. Where tissues are continuous and hydrated, the electrical change can pass. Where cells are damaged, dry, or disconnected, the signal fades and stops. Light subtly shapes this electrical behavior. During the day, photosynthetic activity alters internal ion concentrations, changing how electrical potentials are maintained. Cells tend to be more responsive when light is present. As evening approaches, this responsiveness decreases. Ion movement slows, electrical fluctuations become less frequent, and the internal environment grows quieter. This is not sleep in the animal sense. There is no loss of awareness, but there is a settling, a soft reduction in sensitivity as the day's energy wanes. Temperature also influences the pace of electrical signaling. In warm conditions, ions move more freely through channels, allowing signals to travel with less resistance. In cooler conditions, movement slows, and electrical changes may dissipate before reaching distant tissues. Water supports the entire process. Hydrated cells conduct electrical changes smoothly, allowing signals to move between tissues with relative ease. As water becomes scarce, this continuity weakens. In dry conditions, electrical signals fragment. They travel shorter distances, and fewer cells are affected. Perception narrows as hydration declines. Electrical signaling allows the plant to sense which tissues remain alive. Where the electrical gradient is maintained, cells are functional. Where the gradient collapses entirely, life has ended. This distinction does not require recognition or interpretation. The presence or absence of electrical balance is enough. Not every electrical change leads to further response. Many shifts arise, travel briefly, and fade without triggering anything else. Only signals that exceed certain thresholds open the door to deeper processes. These thresholds are not the same everywhere. Young, actively growing tissues respond to smaller changes. Older, more stable regions require stronger electrical shifts before reacting. Mechanical and chemical disturbances produce distinct electrical signatures. Wind bending a stem creates a short-lived pattern. Chemical irritation produces a longer alteration in voltage. Cells differentiate between these patterns without understanding their source. Electrical signals do not instruct the plant on what to do next. They do not carry plans or outcomes. They simply mark that a condition has changed. Once the ion distributions return to equilibrium, the electrical system resets. Membranes regain their original charge difference. The cell resumes its steady internal state. The electrical layer is the earliest form of information processing in the plant. It comes before chemical messengers, before hormones, before long-term adjustment. It prepares the ground without directing what will follow. And as balance settles back into place, the quiet electrical state holds, leaving the tissue poised for the slower, more specific signals that may soon begin to form and move through the plant, drifting outward as chemistry takes over where electricity has finished, and the story continues into the way these electrical changes interact with the internal rhythms of the plant itself. Within stems and leaves, electrical activity does not remain constant throughout the day. As light intensifies, internal chemistry shifts, subtly altering how charges are held across membranes. Cells become more responsive, not because they are instructed to be alert, but because the conditions for ion movement are more favorable. As sunlight fades, this responsiveness eases. Electrical changes become smaller, less frequent, and more localized. The system grows quieter, conserving energy without shutting down. This daily modulation does not require awareness. It emerges naturally from the interaction between light, chemistry, and membrane behavior. Electrical signals are also shaped by the flow of water through the plant. As water rises through vascular tissues, it carries ions along hydrated pathways, supporting the spread of electrical change. Where flow is steady, continuity is preserved. When flow slows, signals hesitate. They weaken, sometimes stopping entirely before reaching distant tissues. The baththe remains, but the conditions no longer support movement. In this way, electrical signaling reflects the internal state of the plant. It does not override limitation, it mirrors it. Electrical changes can arise from within, not only from external disturbance. Growth itself alters membrane tension. As cells expand, divide, or change shape, ion distributions shift slightly, creating local electrical variation. These growth-related signals are subtle. They do not spread far. They remain close to their origin, adjusting nearby tissues without broadcasting change. This allows development to proceed quietly, without triggering defensive or stress-related responses. Electrical signaling also participates in orientation. As stems bend toward light or roots grow downward, mechanical strain alters ion flow along specific tissues. This creates electrical differences that reinforce direction, helping the plant maintain alignment with gravity and light. The process is gradual. Direction is refined over time, not corrected suddenly. Electrical patterns can overlap. Multiple small changes may exist at once, each confined to its own region. They do not merge into a single message. They coexist. Because these signals fade naturally, they rarely interfere with one another. Each arises, travels briefly, and dissipates before the next becomes dominant. The absence of accumulation is essential. Without it, the internal environment would become saturated with noise. Stress alters this balance. Prolonged heat, repeated injury, or extended dryness shifts baseline electrical properties. Membranes become slightly more permeable. Ion channels respond more readily. Future disturbances require less force to create electrical change. Responsiveness increases without storing a record of the past. This adjustment does not encode memory. It changes sensitivity. Electrical signals do not travel indefinitely. They are bound by time as well as distance. If conditions are not favorable, the signal fades before completing its path. This ensures that only relevant regions are influenced by a given event. In older tissues, electrical stability is stronger. Membranes resist change. Signals tend to be weaker and shorter-lived. In younger tissues, electrical balance is more flexible. Smaller disturbances produce clearer shifts. This supports growth and adaptation where it is most needed. Electrical signaling does not distinguish between helpful and harmful change. It registers deviation from balance, nothing more. Once equilibrium is restored, the signal disappears completely. No trace remains in the electrical system itself. This impermanence is deliberate. It prevents the buildup of outdated information. Electrical signaling is therefore not a means of decision-making. It does not determine outcomes. It prepares tissue by adjusting readiness. Only when the electrical change persists long enough does the next layer begin to form. Chemical messengers emerge slowly, shaped by the altered internal conditions that electrical signaling has made possible. They carry specificity where electricity carries awareness. For now, the electrical layer completes its role. Balance settles back into place. Ion distributions stabilize. Membranes return to their quiet tension. The plant remains standing, unchanged in appearance, yet subtly prepared, as the first silent language of life gives way to the slower chemistry that follows, and the movement of information continues onward into a moment of stillness where nothing further happens. After the electrical balance has shifted and returned, the plant does not remain altered at the surface. Leaves do not change color. Branches do not move. The forest offers no visible sign that anything has passed. Inside, however, the conditions are slightly different than before. The electrical system has done its work by creating a window of awareness. It has marked a change without defining its meaning. It has allowed tissue to become momentarily receptive. Once that window closes, the electrical state settles completely. Ion channels return to their resting positions. Charge differences stabilize across membranes. The background tension that defines living tissue re-establishes itself. There is no lingering signal, no echo, no trace. Electrical signaling leaves nothing behind. This absence is essential. If electrical changes were permanent, the internal environment would become distorted over time. By fading entirely, each signal ensures that the next moment begins without bias. In this way, the plant experiences each disturbance as new. Electrical signaling does not compare events. It does not weigh them against the past. It does not build a story. It exists entirely in the present. The electrical layer also does not discriminate between sources of change. Whether the disturbance comes from wind, touch, injury, or internal growth, the signal registers deviation from balance alone. Meaning is not assigned at this stage. This neutrality allows the system to remain simple. Because electrical signals are short-lived, they cannot carry detailed instructions. They cannot encode complex responses. They cannot coordinate distant regions with precision. Their role is limited by design. They announce that something has shifted, and then they step aside. Only when electrical changes persist or repeat does the internal environment begin to favor the emergence of slower processes. Metabolic pathways adjust their readiness. Certain enzymes become more likely to activate. These changes do not come from electricity itself, but from the altered conditions electricity briefly creates. The electrical layer prepares the ground, but it does not plant anything. This makes it the earliest and simplest form of information handling in the plant. It is not intelligent in the human sense. It is responsive in the biological sense. It allows a living structure to sense itself without awareness, to notice change without understanding, and to remain stable without rigidity. In the forest, this process happens constantly. Thousands of small electrical changes rise and fade every day, most of them unnoticed, most of them inconsequential. They form a quiet background activity beneath the visible calm of leaves and trunks. Nothing in the forest appears busy. Nothing seems alert. And yet, at the level of membranes and ions, life remains gently attentive. Electrical signaling does not make the forest intelligent. It makes it alive. It allows living tissue to remain open to the world without being overwhelmed by it. And when its role is complete, when balance has been restored, it withdraws entirely. What remains is a system ready for something slower, something more specific, something that carries not just awareness, but instruction. As electricity falls silent, chemistry begins to stir, and a different language of the forest prepares to move through the same pathways, carrying information not as charge, but as molecules, drifting patiently from cell to cell. As the electrical layer falls quiet, something slower begins to take shape. Inside plant tissues, tiny molecules start to accumulate. They do not move with the suddenness of charge. They gather patiently, forming gradients that drift rather than pulse. These molecules are not thoughts. They are not messages in the human sense. They are signals, chemical indications that a change has occurred and may require response. In plants, hormones serve as primary chemical signals. They are produced in one place and act in another, carrying information across tissues without awareness or intention. Each hormone influences how cells behave, not by command, but by altering probabilities. Jasmonic acid is one such signal. It forms when plant tissue is damaged, especially by herbivores. As its concentration rises, it shifts cellular activity towards defense, increasing the production of compounds that make leaves less palatable or more resistant. This response does not happen instantly. The molecule must be synthesized, transported, and recognized by target cells. Time passes as chemistry unfolds. Auxin operates differently. It guides growth rather than defense, influencing how cells elongate and divide. By accumulating unevenly across tissues, Auxin helps stems bend toward light and roots grow downward into soil. Its effect depends on where it is concentrated. The same molecule can stimulate growth in one region and inhibit it in another, simply by its distribution. Ethylene stands apart from most plant hormones. It exists as a gas. Once produced, it diffuses freely through the air as well as tissue, moving without the need for transport vessels. This allows ethylene to act both within a plant and beyond it. Ripening fruit releases ethylene into the surrounding air, where it can influence neighboring fruits and leaves. The signal escapes the body of the plant entirely. Some chemical signals do not remain confined to internal pathways. They drift outward, carried by air currents as volatile organic compounds. These molecules are light enough to travel beyond the plant that produced them. Nearby plants can detect these airborne signals. They do not hear or see them. They register their presence through receptors tuned to specific molecular shapes. In this way, a plant can be influenced by chemical changes occurring in another organism nearby. The forest carries information invisibly, suspended in air. Chemical signaling is slower than electrical signaling. Minutes or hours may pass before molecules reach effective concentrations. This slowness is not a limitation. It allows responses to scale with persistence rather than immediacy. Each chemical signal carries a distinct meaning shaped by its molecular structure. Cells respond not to the idea of danger or growth, but to the presence of a specific compound binding to a specific receptor. When a signal molecule binds, enzymes within the cell change their activity. Some reactions speed up, others slow down. Metabolic pathways are adjusted rather than switched on or off. Chemical signals can amplify themselves. A small initial increase in hormone concentration may trigger the production of more signaling molecules, strengthening the response over time. This amplification allows a minor disturbance to produce a significant physiological change if it persists long enough. Plants under attack often release chemical signals before severe damage occurs. Response begins while tissue is still alive, adjusting defenses in advance of further loss. Signals do not move in a single direction. Leaves can send chemical information toward stems. Stems can redistribute signals toward roots. Roots also produce chemical signals of their own. They respond to nutrient availability, soil conditions, and microbial interactions, releasing compounds that influence growth above ground. Not every chemical signal leads to action. Cells respond only when concentrations exceed certain thresholds. Below that level, molecules drift and dissipate without effect. This thresholding prevents constant reaction to background noise. Only sustained or strong signals influence behavior. Low concentrations may result in subtle adjustments. Enzyme activity shifts slightly. Growth rates change imperceptibly. Higher concentrations produce stronger responses. Defensive compounds increase. Growth patterns are altered more dramatically. Not all signals are received. Some molecules pass through tissue without binding to receptors. Others are broken down before they can act. Plants prioritize internal signals over external ones. Chemical changes originating within their own tissues are more likely to trigger response than signals arriving from outside. There is no central processor for chemical information, no location where signals are compared or weighed. Each cell responds locally, based on the receptors it carries and the molecules it encounters. From countless local interactions, coordinated behavior emerges. And as these invisible molecules continue to drift, bind, and fade, the forest fills with a quiet exchange of chemical information, moving slowly through air and tissue, shaping a response without sound. As the language of molecules unfolds further into the way plants interpret these drifting signals without ever translating them into thought. Chemical messages do not arrive as instructions. They arrive as presence. A molecule binds or it does not. And from that simple event, a cascade quietly begins. Receptors, embedded in cell membranes, are shaped to recognize specific compounds. When the correct molecule fits, the receptor changes its form. This change is small, but it is enough to alter what happens inside the cell. Enzymes respond first. Some become more active. Others slow their work. Metabolic pathways shift direction, not abruptly, but gradually, as reactions rebalance themselves. Nothing switches fully on or off. The system adjusts its tendency. Because chemical signals depend on concentration, their effects scale naturally. A faint signal produces only a minor adjustment. A stronger presence alters cellular behavior more noticeably. This allows the plant to respond proportionally rather than absolutely. There is no single reaction to a molecule. There is a range. Chemical signals also take time to disperse. They move through tissues by diffusion, carried by fluids, passing from cell to cell without urgency. As they spread, their concentration decreases. This creates gradients rather than waves. Cells closer to the source experience stronger effects. Distant cells may register nothing at all. Gradients allow direction to emerge without instruction. Growth bends toward light. Resources shift away from damaged areas. No message says where to go. The distribution itself is enough. Some chemical signals amplify their own production. As receptors activate, they can trigger the synthesis of additional signaling molecules. This feedback strengthens the signal over time. Amplification does not guarantee action. If conditions change, production slows and the signal fades. Plants often release defensive chemicals while tissue is still intact. The signal appears early, before damage becomes severe. This timing allows responses to prepare rather than repair. Leaves under attack may alter their chemistry within hours, increasing compounds that discourage feeding. The response spreads slowly, reaching nearby tissues first. Signals can travel inward as well. Leaves send chemical information toward stems, where it influences transport and allocation. From there, signals may continue toward roots. Roots, too, are active signal producers. They release compounds in response to nutrient availability, moisture, and soil conditions. These signals influence growth patterns above ground. Some root signals remain localized. Others travel upward, altering development across the plant. Chemical communication does not distinguish between internal and external space. Volatile compounds released into the air can be detected by nearby plants. These airborne signals carry information beyond the individual organism. A plant exposed to certain airborne molecules may adjust its physiology preemptively. The response is not alarm, it is preparation. Not every molecule drifting through air or tissue is meaningful. Cells ignore most of what they encounter. Only specific shapes bind to receptors, and only when present at sufficient concentration. This selectivity prevents constant reaction. The forest is full of chemical noise. Plants respond only to what persists. Some signals are intentionally short-lived. They break down quickly, ensuring that responses remain temporary. Others last longer, supporting sustained adjustment. Chemical signals can interact. Multiple molecules may influence the same pathway, reinforcing or moderating each other's effects. The resulting response reflects the balance rather than any single signal. There is no hierarchy among these messages. No signal outranks another universally. Context determines outcome. Cells prioritize signals originating within their own tissues. Internal chemical changes are more likely to influence behavior than molecules arriving from outside. This preserves autonomy while allowing awareness. There is no central site where chemical information converges, no place where signals are collected or compared. Each cell responds independently based on the receptors it carries and the molecules it encounters. Coordination emerges from repetition, not control. As signals diffuse and bind, the plant's physiology shifts gradually. Growth patterns adjust. Defensive compounds increase or decrease. Metabolism realigns. These changes do not happen all at once. They unfold over hours, sometimes days, shaped by persistence rather than immediacy. Chemical signaling allows the plant to integrate information over time. Short disturbances fade before causing effect. Sustained conditions leave a deeper imprint. When signals decline, responses ease. Enzyme activity returns toward baseline. Production slows. The system drifts back toward equilibrium. Nothing is stored as a memory. Only the current chemical environment matters. In the forest, these molecular exchanges are constant, invisible, silent, and slow. They move through leaves, stems, roots, and air, weaving a quiet layer of communication beneath the visible stillness. The forest does not speak. It exhales information. And as these chemical signals continue to rise, spread, and dissolve, the language of molecules grows richer, preparing the living system for the deeper patterns of timing and response that unfold over longer scales as the story of forest signals drifts onward into a gentle quiet where chemistry finishes its work. As chemical concentrations rise and fall, the plant does not rush to resolve them. It allows signals to linger just long enough to matter, then lets them dissolve. Molecules break down. Receptors return to their resting shapes. Enzymes ease back toward baseline activity. Nothing announces that the message is over. It simply fades. Chemical signaling does not aim for permanence. Its purpose is adjustment, not transformation. Once conditions shift again, the chemistry shifts with them. Some signals are produced only briefly, designed to disappear quickly. Others persist longer, supporting sustained change, while circumstances remain stable. The lifespan of a molecule shapes the duration of its influence. This impermanence prevents overload. Without it, tissues would remain locked in past responses. Instead, the plant stays fluid, capable of responding anew. Chemical signals also interact with space. Where molecules accumulate densely, responses are stronger. Where they thin out, effects diminish. Distance matters, but not in straight lines. Transport pathways guide diffusion. Vascular tissues carry molecules farther than open space alone would allow. Leaves, stems, and roots each shape how signals move through them. Even so, no signal reaches everywhere. Every chemical message has limits. Its reach is shaped by concentration, breakdown rate, and the structure of the tissue it enters. Because of this, responses remain localized. Defense rises where damage occurs. Growth adjusts where light or gravity demands it. The plant never becomes uniformly altered by a single signal. Variation is preserved. Chemical signaling does not create unity by force. It creates coordination by overlap. Multiple signals may be present at once, each influencing different aspects of physiology. Growth hormones coexist with stress signals. Defense compounds rise while development continues elsewhere. The plant does not choose between them. It expresses the balance that emerges. Not all cells respond in the same way. Receptor presence varies. Sensitivity differs. The same molecule can have different effects in different tissues. This diversity allows specialization without central planning. Roots respond as roots. Leaves respond as leaves. Chemical signals are therefore not commands sent from one part of the plant to another. They are conditions created and shared. When those conditions change, responses change with them. Some airborne signals dissipate before reaching another organism. Others travel far enough to influence neighbors. Whether they matter depends on timing, distance, and concentration. A molecule released into the air may never be received, or it may arrive at exactly the right moment to alter a nearby plant's chemistry. Reception is always conditional. Plants do not listen constantly. They are not tuned to every molecule in their surroundings. They respond only when internal and external conditions align. This selectivity preserves autonomy. The plant remains responsive without being reactive. There is no memory stored in the chemical signal itself. Once the molecule is gone, its influence ends. Only structural or genetic changes persist, and those arise slowly. Chemical signaling bridges the gap between immediate sensing and long-term adjustment. It allows the plant to respond within hours rather than moments, and to integrate information over time rather than instantaneously. In the forest, this chemical language flows continuously. It moves through tissues, across air, between organisms, invisible, silent, and patient. Nothing about it demands attention. Nothing about it seeks to be understood. And yet, through these drifting molecules, plants coordinate growth, prepare defenses, and adjust to their environment with remarkable subtlety. No words are spoken, no messages are sent. Only chemistry unfolding at its own pace. As these signals fade and balance returns, the forest remains still on the surface, while beneath that calm, the quiet work of molecular communication completes its cycle, leaving the living system gently aligned with the present moment, ready for the slower rhythms of time itself to take hold. As the journey continues, beyond chemistry, into the long, patient waves through which plants experience time. Time inside a plant does not pass the way it does for animals. There is no urgency here, no rapid correction, only slow waves of response unfolding at a pace shaped by biology rather than expectation. When a signal begins to move through plant tissue, it does not arrive in seconds. Minutes may pass before the first changes become measurable, sometimes much longer. This delay is not a failure to respond. It is part of the response itself. Plants experience change through gradual physiological waves. Electrical shifts soften into chemical gradients, and those gradients influence processes that take time to express. Nothing snaps into place. Some reactions require hours before their effects become visible. Enzymes adjust slowly. Metabolic pathways realign step by step. The outcome emerges only after the system has had time to settle. This pace allows the plant to filter its environment through duration. Short-lived disturbances fade before provoking deep change. Only signals that persist long enough become meaningful. Biological time in plants is governed by internal rhythms. Circadian cycles shape how tissues respond, determining which processes are available at different times of day. The same signal can produce different outcomes depending on when it arrives. Plants distinguish day from night without awareness. Light alters internal chemistry, shifting sensitivity across tissues. During daylight, responses favor growth and energy capture. In darkness, activity turns inward, supporting maintenance and repair. Light does not simply trigger reactions, it modulates timing. Signals received at dawn unfold differently than those arriving at dusk. The clock inside the plant shapes interpretation without interpretation. Stress does not vanish when conditions improve. Its effects linger, embedded in slow physiological changes. After heat, drought, or repeated disturbance, the system takes time to return to baseline. This persistence is not memory in a narrative sense, it is inertia. Physiological waves continue to travel even after the initial signal has faded. Their momentum carries influence forward, smoothing abrupt transitions. Because of this, plants do not panic. They do not react explosively to sudden change. They absorb disturbance and respond proportionally, spreading adjustment across time. Delay becomes protection. Responses are often intentionally postponed. By waiting, the plant reduces the risk of reacting to conditions that may quickly reverse. Time itself acts as a filter. Slow waves of response conserve energy. Rapid reactions are costly. Gradual adjustment allows resources to be allocated carefully, avoiding unnecessary expenditure. Stability is always favored over speed. Plants prioritize remaining functional over reacting quickly. A delayed response that preserves structure is more valuable than a rapid one that risks imbalance. In this way, time becomes part of the signal. Not just what happens, but how long it happens shapes outcome. Signals that persist alter gene expression. They encourage deeper, longer-term adjustments. transient signals remain at the level of physiology, adjusting function without rewriting underlying patterns. Duration determines depth. Short signals lead to reversible changes. Long signals reshape behavior over extended periods. This distinction allows plants to remain flexible. They adapt without committing prematurely. All of this unfolds within what might be called deep time. Plants live on scales that stretch beyond immediate events. Seasons matter more than moments. Cycles outweigh incidents. A tree does not experience a single hot day as a crisis. It experiences it as part of a pattern. Slow waves integrate information across hours, days, and months. They smooth sharp edges. They favor continuity. From the outside, this may look like inaction, but within, the system is constantly adjusting, measuring time not by clocks, but by persistence. Change arrives gently. Response follows carefully. And as these slow physiological waves continue to move through living wood, shaping reaction with patience rather than urgency, the plant settles into its own temporal rhythm, breathing in time, breathing out response, and allowing the present moment to stretch long enough for meaning to emerge. As the story of forest time drifts onward into the way these slow rhythms shape every response the plant allows itself to make. Inside living wood, time is not a background condition. It is an active element. Processes unfold only when enough time has passed for them to matter. Signals do not ask whether the plant is ready. Readiness is determined by rhythm. Circadian cycles open and close windows of responsiveness. At certain hours, tissues are primed to adjust growth. At others, they resist change, holding form steady through the night. This rhythm allows the same signal to be received differently without confusion. A chemical cue arriving at noon may support expansion. The same cue arriving at midnight may pass without effect. Nothing is misread. Timing is part of interpretation. Because responses are slow, the plant avoids abrupt transitions. There is no sharp shift from calm to alarm, no sudden surge of activity. Instead, change accumulates gently. Each small adjustment builds on the last, forming a gradual arc rather than a spike. This gradualism protects structure. Cells are not pushed beyond tolerance. Resources are not drained suddenly. Energy is spent carefully, spread across time, rather than concentrated in moments of urgency. Plants do not attempt to correct every deviation immediately. They allow conditions to persist before committing to response. A brief drought does not trigger drastic adjustment. A sustained one eventually does. Time itself becomes a measure of reliability. Physiological waves continue even after initial signals have faded. Metabolic changes linger, slowly tapering off rather than stopping abruptly. This creates continuity between states. The plant never snaps back to baseline. It eases back. This easing reduces stress. It prevents oscillation between extremes. Stability is preserved through patience. Because responses are delayed, plants rarely waste energy on fleeting events. A shadow passing overhead does not provoke change. A single gust of wind does not alter growth. Only patterns that repeat or persist shape behavior. Slow waves also coordinate distant tissues without direct communication. As changes unfold gradually, different regions have time to adjust in harmony. No part of the plant races ahead of the rest. The whole organism moves together, not by instruction, but by shared pace. This shared pace creates resilience. When conditions fluctuate, slow responses dampen extremes. The system absorbs variation rather than amplifying it. In this way, slowness becomes strength. Signals that remain present long enough begin to influence gene expression. These changes are not immediate. They emerge only after prolonged exposure to consistent conditions. Gene expression adjusts slowly, committing the plant to deeper adaptation only when necessary. This protects against premature change. Short-lived signals never reach this level. They remain at the surface, altering physiology without rewriting internal patterns. The plant maintains flexibility by separating fast change from slow change. Immediate conditions influence function. Persistent conditions influence form. Time defines the boundary. Living in deep time means responding to seasons rather than moments. Growth reflects accumulated experience, not isolated events. A tree carries the imprint of years, not hours. Heat, cold, light, and darkness leave traces only when they endure. Passing extremes are absorbed and released without lasting effect. From the outside, this may look like indifference, but it is discernment shaped by time. Plants do not hurry because there is nowhere to rush. Their lives are measured in cycles that extend far beyond immediate survival. Roots grow slowly because soil does not change quickly. Wood forms gradually because structure must last. Every response is calibrated to longevity. Slow waves allow the plant to remain aligned with its environment over long periods. They integrate information across days, weeks, and seasons. They smooth noise into signal. The present moment stretches. It becomes wide enough to include context. In this expanded present, reaction becomes adjustment. Adjustment becomes alignment. Nothing about this process seeks efficiency in the human sense. It seeks endurance. The forest breathes in long intervals. Growth and rest alternate without urgency. Change arrives, settles, and is absorbed. Time is not something plants endure. It is something they use. And within this slow unfolding, signals find their place. Some fade quickly, some linger. Only a few shape the future. As these slow waves continue to move through the trunks and leaves, carrying the imprint of duration rather than speed, the forest settles into its own tempo, one that stretches beyond moments and into seasons, beyond seasons and into years, reminding us that life does not always respond by acting quickly, but by waiting long enough for the right response to become clear. As time stretches and responses settle into rhythm, something quieter remains behind. Not a signal moving through tissue, not a wave still traveling, but a trace. When a plant experiences stress, the moment does not vanish entirely when conditions improve. Something subtle stays within the living structure, woven into cells, rather than stored as a memory. This trace is not a recollection. It is not an image or an event replayed. It is a biological mark, a small adjustment left behind by experience. Stress alters the internal state of cells. Heat, drought, repeated disturbance, or chemical imbalance change how genes are expressed. Even when the DNA itself remains unchanged, the sequence stays the same. Its activity does not. During drought, for example, certain genes become more active, while others quiet down. Proteins related to water conservation, root growth, and stress tolerance increase. These changes help the plant survive the present moment. When water returns, the immediate crisis ends. Growth resumes. Leaves recover. But the internal balance does not return exactly to where it began. The plant now responds differently to similar conditions. If drought returns, the reaction is often faster. The system adjusts with less delay, as if it has already prepared. This is not learning in the conscious sense. It is memory without awareness. The memory does not reside in a brain. It does not travel as a signal. It exists within the structure of the cell itself. Chemical tags known as epigenetic marks attach to DNA or the proteins that package it. These marks influence which genes are more likely to be expressed and which remain silent. They act as annotations rather than edits. Epigenetic marks do not encode events. They encode tendencies. A gene marked by past stress may activate more readily in the future. Another may remain suppressed until conditions shift dramatically. These marks can persist long after the original stress has passed. In some cases, they remain through multiple seasons, shaping response across time. The plant does not remember what happened. It remembers how to respond. This form of memory is entirely physiological. It does not involve thought, reflection, or awareness. No part of the plant revisits the past. Instead, experience reshapes readiness. Stress memory is not precise. It does not preserve details. It does not distinguish between exact causes, but it is sufficient. A plant that has endured moderate stress often becomes more resilient. Its responses are calibrated by prior exposure, allowing it to tolerate similar challenges more effectively. This resilience emerges from adjustment, not anticipation. Mild stress can strengthen future response. It fine-tunes thresholds and speeds up activation. The system becomes less reactive, yet more prepared. Severe stress leaves a different imprint. When damage exceeds tolerance, cells may be permanently altered or lost. Memory, in this case, becomes limitation rather than advantage. Not all stress produces beneficial memory. Intensity matters. Stress memory is also temporary. Epigenetic marks can fade as cells divide or conditions stabilize. Over time, unused adjustments dissolve. The plant does not cling to the past indefinitely. It releases it when it is no longer useful. Memory does not travel as thought from one part of the plant to another. It is carried forward as tendency, as altered probability. Cells shaped by experience behave differently, and as they divide, they pass this altered behavior on. In this way, memory spreads slowly, not through signals, but through growth. The plant does not store history. It stores responsiveness. And as these quiet imprints settle deeper into living tissue, the forest carries within itself not stories of what has happened, but gentle adjustments shaped by what has endured, allowing the past to sleep softly inside the present. As the nature of stress memory continues to unfold into the way these quiet imprints shape the future without ever becoming thought. Stress memory does not guide behavior through recollection, it shifts the baseline from which behavior emerges. The plant does not remember drought. It becomes slightly different after it. Cells that carry epigenetic marks behave with altered sensitivity. When conditions resemble past stress, thresholds are crossed sooner. Responses begin with less delay, not because the past is recalled, but because readiness has changed. This readiness is unevenly distributed. Not every tissue carries the same imprint. Regions most affected by stress tend to retain the strongest marks. Leaves exposed to prolonged dryness may adjust differently than shaded tissues. Roots experiencing nutrient scarcity may alter their growth patterns more persistently than stems. Memory, in this sense, is local before it is systemic. As cells divide, these altered tendencies can be passed forward. New cells inherit not an event, but a disposition, a likelihood of responding in a certain way. This inheritance is not guaranteed. Epigenetic marks can be diluted, overwritten, or erased as growth continues. Memory fades when it is no longer reinforced. Because of this, plant memory is fluid. It strengthens with repetition. It weakens with time. The plant does not hold on to stress that no longer matters. Adaptation is always provisional. Stress memory does not aim for accuracy. It does not need to distinguish between slightly different forms of hardship. It only needs to bias response toward survival. This makes it imprecise but effective. A plant shaped by past drought may respond similarly to heat or salinity. The memory is not specific. It is pragmatic. This generalization allows the organism to cope with uncertain environments. Exact recall would be unnecessary. Flexibility is more valuable. Because memory is encoded biologically, not cognitively, it cannot be accessed or suppressed. The plant does not choose when to remember. The memory expresses itself automatically through altered response. This expression is subtle. Growth rates change slightly. Stomata open or close with different timing. Hormonal signals rise more quickly or more slowly than before. From the outside, these differences are barely visible. Yet over time, they shape survival. Stress memory does not create foresight. It does not allow the plant to anticipate events before they occur. It only modifies how the present is met. In this way, the plant remains anchored in now. Repeated mild stress often leads to increased resilience. The system learns to tolerate variation without overreacting. Responses become measured, conserving energy while remaining effective. This resilience is not courage, it is calibration. When stress exceeds tolerance, memory becomes damage. Cells may lose function. Tissues may fail to recover fully. In such cases, the imprint left behind limits future response rather than enhancing it. Memory does not always confer advantage. The difference lies in intensity and duration. There is a narrow range where stress informs adaptation without overwhelming structure. Outside that range, survival narrows. Stress memory also interacts with time. Marks that are not reinforced gradually fade. As seasons pass without similar conditions, cells return toward their original state. This erasure is not loss, it is renewal. By forgetting what no longer applies, the plant remains adaptable. It avoids being shaped permanently by a world that has already changed. There is no archive of experiences, only the present configuration of the system matters. Stress memory does not travel through signals the way information does. It moves through growth, division, and replacement. Old cells carry old tendencies. New cells reflect current conditions. Over time, the plant becomes a record of its environment, written not in stories, but in structure. This record is always incomplete, always shifting. The plant does not know what it has endured. It does not compare past and present. It does not judge improvement or decline. It simply responds from where it is. And where it is has been shaped gently and invisibly by what has passed. As these quiet imprints persist and dissolve across tissues and seasons, stress memory reveals itself not as remembrance, but as adjustment carried forward, allowing life to remain flexible without reflection, resilient without awareness, and open to the present moment. As the final layers of this silent memory continue to settle into a state that feels less like remembering and more like becoming. Stress memory does not sit apart from the plant's ongoing life. It blends into structure indistinguishable from growth itself. Over time, what was once a response becomes simply the way the organism exists. Cells shaped by experience are replaced by new ones. Some carry forward altered tendencies, others do not. The balance shifts gradually, guided by current conditions rather than past events. Because of this, memory is never complete. It is always partial, always provisional. Only what remains useful is preserved. The plant does not protect memories, it protects function. As seasons pass, epigenetic marks that are no longer reinforced begin to fade. Cell divisions dilute them. Enzymatic processes remove them. Slowly, quietly, the system resets. This forgetting is not loss. It is adaptation in reverse. By releasing outdated adjustments, the plant avoids becoming trapped by former environments. It remains open to what is happening now. Stress memory, therefore, exists on a spectrum. Some imprints last days, others last months. A few persist across multiple seasons. None last forever. There is no place where these memories are gathered, no archive, no center. Each cell carries its own version of the past, expressed only through how it behaves in the present. From the outside, the plant looks unchanged. The trunk does not reveal its history. The leaves do not display what they have endured. And yet, the internal landscape has been shaped. Roots grow with slightly altered priorities. Leaves regulate water with subtly different timing. Hormonal signals rise and fall along paths influenced by what has come before. These differences are small, almost invisible, but across time, they matter. Stress memory does not give the plant foresight. It does not predict. It does not plan. It simply narrows the distance between disturbance and response. The next time conditions shift, the plant meets them from a place already adjusted, not prepared in advance, but shaped by what has endured. This shaping does not create certainty. It creates tendency. The plant never knows whether a response will be sufficient. It only responds from where it stands. In this way, stress memory supports survival without guaranteeing it. It offers resilience, not protection. The forest is full of these quiet imprints. Every tree carries within it traces of drought, heat, wind, and scarcity. None of these traces are stories. They are adjustments. Together, they form a living archive written not in recollection, but in form. This archive is constantly rewritten. Pages are added. Pages are erased. Nothing is permanent. The forest does not dwell on what has passed. It does not replay experience. It does not cling to survival strategies that no longer apply. Instead, it allows the past to sleep gently inside the present, influencing response without demanding attention. Memory, in plants, is not about holding on. It is about letting experience change structure just enough to matter and no more. There is no awareness of having learned, no recognition of having adapted, only life continuing, slightly altered, moment by moment. As stress memory settles into this quiet role, never spoken, never recalled, never fully retained, it completes its work by dissolving back into physiology, leaving behind not a record of the past, but a living system subtly shaped by what it has endured, ready to respond again without remembering, as the forest carries forward not stories, but the gentle consequences of time. Not everything that touches a plant deserves a response. Most changes pass through unnoticed. They arrive, brush against living tissue, and disappear without leaving a trace. This restraint is not indifference, it is design. Plants operate with thresholds. A signal must reach a certain intensity before it matters. Below that level, it is treated as background. Weak signals are ignored. Small fluctuations in light, brief touches, momentary dryness, all pass beneath the line of response. The plant remains still. This selectivity protects energy. Every response has a cost. Producing enzymes, altering growth, activating defense require resources that cannot be wasted. By ignoring what is too small, the plant preserves itself. Thresholds are not rigid. They shift depending on tissue, age, and condition. What matters in one part of the plant may be irrelevant in another. Young tissues tend to be more sensitive. Their thresholds are lower. They respond to subtle cues that guide growth and orientation. Older tissues are more stable. Their thresholds are higher. They resist change, preserving structure built over time. Different species set these thresholds differently. Plants adapted to harsh environments tolerate greater variation before responding. Those in stable conditions may react to smaller changes. There is no universal setting. Each organism tunes itself to its world. Once a signal crosses a threshold, response does not explode outward. It unfolds carefully, moderated by feedback. Negative feedback acts first. As a response begins, counteracting processes rise to limit it. Growth slows itself. Defense dampens its own intensity. This prevents overreaction. The plant does not swing wildly between states. Stability remains the goal. Positive feedback plays a different role. When conditions persist, initial responses reinforce themselves, A signal that remains strong encourages its own continuation. This allows important information to spread, but even here, limits remain. Feedback does not create urgency. It creates persistence. The system is designed to avoid extremes. No single signal overwhelms the whole organism. Responses remain proportionate to conditions. Information noise is filtered continuously. Random fluctuations cancel themselves out. Only consistent signals shape behavior. This filtering is constant and automatic. No assessment is made. No choice occurs. Thresholds and feedback operate together, shaping response without awareness. The plant does not ask whether it should respond. The system responds only when balance is disturbed enough to require adjustment. There is no instant decision. Time always participates. Signals must be strong enough and last long enough. A brief spike fades before crossing the threshold. A sustained change gradually accumulates influence. This temporal requirement prevents reaction to transient events. Only patterns that endure matter. Feedback loops slow everything down. They stretch response across time, smoothing sharp edges. The plant settles into adjustment rather than reacting abruptly. This slowness is not hesitation, it is control. The system behaves like a natural regulator. Inputs arrive, outputs adjust, balance is restored. No central processor oversees this process. No part of the plant issues commands. Each cell responds locally, guided by shared rules rather than instruction. Coordination emerges from repetition, not authority. Thresholds ensure silence when nothing matters. Feedback ensures moderation when something does. Together, they allow the plant to remain stable in a world that is always changing. And as these invisible limits quietly govern response, the plant continues to move not toward action or inaction, but toward equilibrium, as the deeper patterns of feedback begin to unfold into the way balance is restored without ever being declared. When a response begins, it immediately creates the conditions for its own moderation. As activity rises, opposing processes rise with it. Growth stimulates restraint. Defense invites limitation. This negative feedback is quiet and constant. It does not stop responses outright. It shapes their edges. Without it, even small disturbances could escalate. With it, change remains contained. As water loss increases, stomata begin to close. As they close, water loss slows. The very response reduces the signal that caused it. This loop continues until balance is approached again. Negative feedback keeps systems near their preferred state, not fixed, not rigid, but stable. Positive feedback works more selectively. It appears only when conditions persist beyond brief fluctuation. A signal that remains present strengthens the response that produced it. This reinforcement allows important adjustments to spread gradually through tissue. But even here, limits apply. Positive feedback does not remove restraint. It works within boundaries set by negative loops. Together, these opposing forces prevent extremes. Responses grow only as much as conditions justify. Because of this interplay, plants rarely experience runaway reactions. There is no equivalent of panic, no sudden cascade that overwhelms the organism. Instead, response unfolds like a settling wave. Feedback loops also operate across time. As conditions improve, responses unwind slowly rather than stopping abruptly. Activity tapers off. Balance is approached gently. This gradual return prevents oscillation. The system does not overshoot its target. Everything moves toward equilibrium, not efficiency. Every response carries cost. Energy spent on defense cannot be spent on growth. Resources allocated to repair are unavailable elsewhere. Feedback ensures the costs are weighed implicitly. Excessive response is discouraged, not by decision, but by consequence. Thresholds and feedback together form a filter. They decide what matters by refusing to respond to what does not. This filtering is continuous. Signals arrive constantly. Most dissolve before crossing the line. The plant is never overwhelmed because it is never fully open. Noise is everywhere. Shifting shadows, passing wind, transient dryness. Thresholds keep this noise from becoming information. Only when change persists does it become meaningful. Feedback also varies across tissues. Young leaves may amplify signals more readily. Older wood dampens them. This creates a gradient of responsiveness. Sensitive regions adapt quickly. Stable regions hold form. The plant does not need to coordinate this difference. It arises from structure. Different species tune feedback differently. Fast-growing plants allow quicker shifts. Slow-growing species resist change more strongly. Each system is calibrated to its environment. There is no correct setting, only what survives. Feedback loops do not rely on awareness. They require no perception of outcome. They simply connect cause and effect. When a process begins to exceed usefulness, opposing forces rise automatically. When a response remains insufficient, reinforcement builds. No one directs this. No center oversees it. Control is distributed across the system. Because responses are always moderated, there is no instant decision point. Nothing switches fully on or off. Everything exists in degrees. This gradation allows fine adjustment. Small changes produce small responses. Large, sustained changes produce larger ones. Time remains essential. Signals must be strong enough and persistent enough. A brief spike may cross intensity threshold, but fail duration. A long, mild signal may accumulate influence slowly. Only when both conditions align does response unfold fully. This requirement protects the plant from reacting to coincidence. Patterns must endure to matter. Feedback loops stretch these patterns across time. They smooth sudden inputs. They reduce volatility. The plant behaves like a system constantly tuning itself. Inputs arrive, outputs adjust. Balance is approached again and again. There is no end state, only continual regulation. And as these loops continue their quiet work, guiding response without command or awareness, the plant remains poised between sensitivity and restraint, allowing change to be felt without being overwhelmed by it, as the principles of thresholds and feedback settle deeper into living tissue, shaping behavior that moves always towards balance, as the system's final quiet adjustments begin to drift into a state where balance no longer needs to be maintained actively, because it has been rebuilt from within. When thresholds have been crossed and feedback has done its work, response gradually dissolves. not abruptly, not all at once. Processes unwind in reverse order. Signals weaken. Enzyme activity slows. Resource allocation eases back toward baseline. Nothing declares completion. There is no moment when the plant stops responding. The system simply settles. Feedback loops guide this settling gently. As conditions improve, reinforcing signals lose strength. Counteracting processes regain influence. Response diminishes because it is no longer supported. This prevents the plant from overshooting stability. It does not rush to return to a previous state. It approaches equilibrium slowly, allowing tissues to readjust without strain. Because thresholds must be crossed again to restart response, the system remains calm during minor fluctuations. Once balance is near, only significant change can disturb it again. This hysteresis protects continuity. The plant does not oscillate between states. It rests where it arrives. There is no memory stored in these loops. They do not recall what came before. They simply respond to present conditions according to their rules. Feedback is not wisdom. It is structure behaving predictably. Yet from this predictability, something remarkable emerges. The plant appears composed, unhurried, unaffected by most of what passes around it. This appearance is not passivity. It is regulation working continuously beneath the surface. Thresholds ensure that silence is the default. Feedback ensures that action, when it occurs, remains measured. Together, they create a system that rarely needs to react strongly, because it rarely allows imbalance to grow unchecked. There is no urgency because urgency is unnecessary. The plant does not seek efficiency, it seeks persistence. By responding only when signals are strong enough and long enough, it avoids waste. By moderating every response through feedback, it avoids damage. Nothing is optimized for speed. Everything is shaped for endurance. This endurance does not come from foresight or planning. It comes from rules embedded in physiology. Rules that require no awareness. Rules that operate everywhere at once. Each cell follows the same principles. Each region participates without instruction. Control is not centralized. It is shared. When conditions shift, the entire system adjusts together, not because it is told to, but because every part responds according to the same constraints. Thresholds define when change matters. Feedback defines how much change is allowed. Time ensures that only persistent signals pass through both gates. This triad, threshold, feedback, and duration, forms the final layer of signal intelligence in the forest. It is not about deciding. It is about balancing. The forest does not weigh options. It does not choose between paths. It settles. Again and again, it settles. Through drought and rain, heat and cold, abundance and scarcity, these quiet systems continue their work, filtering, moderating, stabilizing. Nothing about them seeks attention. Nothing about them announces control. And yet without thresholds and feedback, the forest could not remain whole. As these final adjustments fade into equilibrium, the living system returns to its quiet default state, responsive but restrained, sensitive but stable, holding itself gently within the bounds that allow life to continue without urgency, without excess, and without awareness. completing the long, patient story of forest signals, where intelligence is not found in decisions or thoughts, but in the soft, self-regulating balance that allows the forest to remain at rest. There is nothing left to follow now, no signals to trace, no patterns to hold in mind. The forest has done its quiet work. Inside the trees, electrical currents have softened and released. Molecules have drifted back into stillness. Thresholds have closed, not with force, but with rest. And you can rest too. If your eyes are still open, you don't need to close them yet. If they are already closed, let them remain that way. There is nowhere else to go. Allow your breathing to slow, just a little. Not deeper, not controlled, simply slower, as if it has all the time it needs. The forest does not hurry through the night, neither do you. Roots stay in the dark without effort. Leaves hold the cool air without moving. Time stretches wide and quiet. You don't need to understand what you've heard. You don't need to remember it. The signals have already passed. All that remains is calm, steady, and safe. Let your body follow the same rhythm as the forest now. Slow, patient, at rest. There is nothing more to do. Good night.